we'll wait just another minute while folks join, and then I will get started.
Will we be able to see them at any point? Or is it just one way?
It is just a one-way screen, unfortunately. Alrighty, why don't we go ahead and get the show on the road. All right, welcome everyone, and thank you for joining us here at New Mexico Tech. And today we are going to talk about the COVID-19 vaccines and give you some information about the historical perspective on vaccination in the United States and around the world, how the vaccines work, not only just at a surface level, but also some of the molecular biology that goes into making an mRNA vaccine work, what's in them, that sort of thing. And then we're also going to talk about safety and efficacy of the vaccines and how those vaccines can impact you, both protecting from COVID as well as a lot of worry about allergic responses and that sort of thing. So let me do some introductions. First, my name is Dr. Joel Charbrough. I'm a new assistant professor here at New Mexico Tech. I just started in January, in fact. Essentially, with the goal of this panel is to provide information from people that the tech community and Socorro at large can trust to give them, to the best of our knowledge and ability, the information that we have. My motivation specifically for being involved, I mean, when you're a new assistant professor, there's a lot thrown at you, right? But this was really important to me to make sure that everybody got the right information. I think in the last 6 months, we have all seen the devastating effects of bad information can have on society. And essentially seeing people turn down the vaccines was really devastating to those of us in the biology community and in the medical community. So to that end, we have a group of people who have expertise on this topic, and I thought it worthwhile to get them to you and provide them to you. I myself am not an immunology or virology expert by any means. I just am a moderator for today's discussion. So with that, I would like to introduce our panelists. So first, let me start off with Dr. Tom Kieft. Dr. Kieft is an environmental microbiologist who investigates the ecology and biogeochemistry of microbes in extreme environments. And he also teaches a course, Infection and Immunity, here at New Mexico Tech that deals with these topics. Dr. Kieft completed a master's degree in biology at New Mexico Highlands University in 1978, got his PhD in biology at the University of Mexico in 1983. He was an assistant research microbiologist in the Department of Plant and Soil Biology at the University of California, Berkeley, from 1983 to 1985. And then in 1985, he joined the Tech faculty and has been here ever since. He is a member of the Deep Life Sciencing Steering Committee for the Deep Carbon Observatory. And in his research, Dr. Kieft has investigated the deep biosphere in South African gold mines and developed new methods for detecting pathogenic microorganisms and microbial toxins. In his recent work, he's been focusing on uncovering relationships between the abundance and diversity of microbes that live in association with animals and animals' mass. All right, welcome, Dr. Kieft. I don't think we can hear, hopefully everybody's applauding. Next, and Dr. Kieft will provide some historical context for vaccination efforts, both in the US and around the world. Next is Dr. Snezhna Rogel. Dr. Rogel received her bachelor's degree in physics with a concentration in astronomy from The Ohio State University, and her PhD in biochemistry from the Boston University School of Medicine. And during her 4-year postdoc at MIT's Whitehead Institute with Dr. Robert Weinberg, she participated in the cloning of the first tumor suppressor gene, RB, and showed that subcellular localization of growth factor FGF plays a critical role in tumorigenicity and metastasis. After a year of angiogenesis research with Dr. Michael Klagsbrunn and Dr. Judah Folkman at Harvard Medical School, she worked for 4 years in Bangalore, India, where she studied the brain infiltrating potential of cysticercosis causing parasite T. solium. Upon her return to the US, she studied the molecular mechanisms of adhesive interactions between leukocytes and endothelium at the University of New Mexico in the Department of Pathology. And she has been at New Mexico Tech for the past 22 years, where she is now a full professor. During her 8 years as department chair, she helped engender establishment of, she helped engender establishment of 2 interdisciplinary degree programs, the Biomedical Sciences BS program and the Biotechnology PhD program. While her research at New Mexico Tech has been broadly interdisciplinary, ranging immunosuppressive effects of pesticides and environmental pollutants, biomimetic CO2 sequestration to the development of molecular biosensors, from nanotechnology to the development of antimicrobial materials, her prevailing focus over the past 10 years has been on the development of novel anti-cancer, antibacterial, and anti-parasitic drugs. Her recent explorations into the realm of drug entrepreneurship and have been rewarded by licensing and commercialization of a novel antimicrobial material with anti-SARS-CoV-2 properties. And for all the many top-notch publications and patents, she is by far the most proud of all this work by mentoring and the research training her hundreds of New Mexico Tech undergraduates and graduates have received over the years, including 40 graduate students. Dr. Rogel will focus on the molecular biology and mechanisms of the vaccine and how it works to produce an immune response in our bodies. Next is Dr. Danielle Turner. Dr. Turner received her Bachelor's of Science degree and Master of Science degree in biology and her PhD in biotechnology from New Mexico Tech. During her time as a postdoc fellow at New Mexico Tech with Dr. Rogel, she participated in the development of novel anti-cancer, antibacterial, and anti-parasitic drugs. She has also immersed herself into the realm of drug entrepreneurship and has successfully licensed and commercialized a novel antimicrobial material with anti-SARS-CoV-2 properties. Dr. Turner also served as an interim COVID-19 testing coordinator at New Mexico Tech in 2020. And she is currently a medical student at the University of New Mexico and is serving both as a postdoc in the New Mexico Tech biology department and taking med school classes online until they resume in-person activities. And Danielle finds most of her joy in the trials and tribulations associated with laboratory work. So Dr. Turner will be focusing on the safety and efficacy data of the available vaccines. Next, we have Dr. Bryce Chakarian, who is the Jeffrey Michael Gervetzian Endowed Professor of Biomedical Research Excellence and Vice Chair of the Department of Molecular Genetics and Microbiology at the University of New Mexico School of Medicine. So Dr. Chakarian is here today to discuss the vaccine molecular biology and mechanisms, as well as serve as an ad hoc panelist where he will contribute to the discussions ongoing from our fellow panelists. We may be joined by Dr. Steve Stewart at some point. He's currently on call. Dr. Stewart is a internist and family medicine physician at Presbyterian Healthcare Services here in Socorro, and he will join if he can. So with that, I'd like to say welcome to all our wonderful panelists. Thank you for taking the time out of your days to contribute to this effort. And without any further ado, I'd like to turn it over to Dr. Kieft for an introduction to vaccine history and efforts over the past 100 years. Thanks.
All right, thanks, Joel. I'm going to share my screen and use a few PowerPoint slides here. All right, can you see my title slide here showing somebody getting vaccinated? All right, so I'm going to give a historic perspective on this, and thanks to Joel, you now know approximately how ancient I am, so maybe that's why I'm the one giving the historic perspective here. I'm going to talk about some history of vaccines, all success stories. There have been few, if any, horror stories in the vaccine realm, and vaccines are a big reason why we in the modern world enjoy such good health. So vaccines have been around a long time, going back perhaps to the 1500s, at least in China, India, elsewhere. Vaccines for smallpox are actually well documented. The first really good smallpox vaccine was credited to Edward Jenner in 1796, who used cowpox, a related virus, to vaccinate people, and that's the source of the word vaccination, because it comes from vaca for cow. Anyway, he's credited with this, and probably the most important thing he did was that he actually collected data and had a controlled test of the vaccine. And you can draw a direct line from Edward Jenner's work to the eradication of smallpox in 1977. First disease to be totally eradicated from the earth, and we're probably going to have a second one in smallpox shortly. Excuse me, in polio, rather. All right, so not quite so long ago, but 100 years ago, diphtheria vaccine was introduced, and there was a rapid drop in the incidence of diphtheria in the United States. And I'm going to show several graphs like this and go through them kind of quickly. Polio. I'm old enough that I remember polio. I remember kids in school a little older than me who had braces on their legs and walked with crutches. I never saw anybody in an iron lung, but there were some of those as well. The polio epidemic had been raging throughout the first half of the 20th century. 60,000 cases in 1952 alone, 3,000 deaths, most of them children, and thousands of children, mostly children, paralyzed. I don't know how they got these girls to smile. These are pretty grim pictures. Vaccination stopped it. The Salk vaccine was distributed widely starting in 1955. I consider that I probably missed out on the polio epidemic because of that, but not by much. And by the way, the Salk vaccine, when it was rolled out and given to millions of people in the US, was probably less well vetted than the Pfizer and Moderna vaccines have already been tested. But it was effective. It worked. It dropped the incidence dramatically. It was followed by another vaccine widely given in 1962. And just about everyone got the vaccines. I'm of an age that people in my generation always question authority. And I still ascribe to that. And you're right to question vaccines, but I think you should come to the conclusion that they are safe and that we're all benefiting from that. It's a government-sponsored project, and clearly not everything that the government has sponsored has worked out well, but in the case of vaccines, yes. And we're about to see polio eradicated worldwide. The numbers here are very low, and they're restricted to a small area in Africa and a small area in South Central Asia. If not for war, this probably would have been eliminated a few years earlier, even within a year or two, no more polio. We don't have to take smallpox vaccines or polio vaccines in the United States anymore. I think that's led to a certain complacency. We reap the benefits without even having to take those vaccines. Measles. I remember when measles was a rite of passage for kids. Measles, mumps, chickenpox, those were the childhood diseases. In some cases, parents would actually send their kids to the home of somebody who with a case just to get it over with. So measles, no big deal, right? Except there's actually a fatality rate of 3 to 5%. So some of those kids getting those childhood diseases were dying. Not anymore because of vaccines. And this is one that we still have to be vigilant about and make sure that kids in the schools get their vaccines. Occasionally there are outbreaks in communities where not enough people are getting vaccinated. And this is nearly my last slide. A lot of information here, but basically it's showing that the incidence of infectious disease has dropped dramatically in 100 years, during the course of the 20th century. And there are multiple factors: water and wastewater treatment, antibiotics, but a big part of it is vaccines. And so we need to be vigilant and keep that up. I've got a couple more slides. I'm going to skip through a couple of these because Snezna, I know, is going to give information on the actual vaccines, what's in them. I don't know if anyone else is showing this, but as of yesterday, 18.4% of New Mexicans have received at least the first shot. That's 360,000 people. Okay? You're not hearing about negative effects that I'm aware of among those 360,000 New Mexicans. It's not enough yet, but we're maybe getting there. And I'll stop there.
All right, thank you, Dr. Kieft. So now what I'd like to do is sort of open it up for discussion about historical perspectives on vaccination and vaccination history. Would any of our panelists care to chime in? If so, go ahead and unmute yourself. Dr. Chakarian.
I was just going to, thanks, Tom, that was a really nice presentation. I was just going to say, I think one of the concerns that people had initially about these new vaccines for the coronavirus was just how quickly they were developed. I think early on in the development of these vaccines, there was a statistic cited, which was 4 years, which was the fastest that we went in approving a vaccine. That would have been the mumps vaccine back in the 1960s. And I think some of the unease that people have about this vaccine is that rapid timeline. And I just wanted to sort of reiterate a few things. How did we get this vaccine so quickly? I think we're going to hear a little bit more about this vaccine in some of the presentations, but it's really a couple of different factors. One factor is that, we have so many advances in molecular biology and immunology, and those advances have really allowed us to respond very rapidly to these sorts of threats. The technologies that we now have at our disposal to develop vaccines are really fantastic, and at least in this case, it's really paid off, and we did get this vaccine quickly. So that's one factor. A second factor is that we actually knew a lot about this virus going in. There have been outbreaks of other related viruses, SARS, the first SARS, MERS virus, and people have been working on vaccines for those other related viruses. And so we had some sense of what sort of immune responses and what sort of vaccine technologies would work. And then the third factor, I think, is just the immediacy of the problem, the fact that we had so many people getting sick and so many people in the scientific community started working on this problem. It was easier to develop all sorts of vaccines. There was a lot of incentives for developing vaccines, a lot of money that the federal government put into developing the vaccines. And then actually it was also easy to test whether the vaccines worked because so many people were getting sick. And that allowed us to get to the clinical endpoints of these clinical trials very, very rapidly. And so, you know, it seems like it was fast, but it's a good thing that it was fast. And it's just this sort of convergence of things that allowed us to go from not even knowing the vaccine, that this pathogen existed, to 11 months later having a vaccine. It's really fantastic.
Thank you, Dr. Chakarian. That's really helpful. And I think especially on, I mean, at least from a basic scientist perspective, like myself, the development of technology is not a new thing. This is stuff that's been coming online for 30 to 40 years. Like RNA sequencing, for example, is fundamental to our ability to create this vaccine, right? To respond to the pandemic in any way, and was developed as part of the Human Genome Project. So completely unrelated technologies that have been harnessed together in this effort. And yeah, I think Dr. Rogel and Dr. Turner can really speak to the all hands on deck moment that this has been in science communities. Would you two care to comment on what it's been like sort of in the scientific community in this historical sort of response to the pandemic?
Certainly. It's absolutely incredible how everybody came together globally and published their results. I mean, that really made a difference. The sharing of data, in addition and on top of the preparation that came from working on other potential messenger RNA vaccines, for example, over just the last decade and most so over the last 5 years. It's been an incredible explosion of you know, from my perspective, scientists coming together to create this based on a technology without which this would not have been possible, technology that had been developed, as you mentioned, over the last few decades.
Yeah, I second Dr. Rogel. It's been amazing, just the global collaboration that has occurred to create these vaccines. And she covered every other point really perfectly and thoroughly. So I don't have anything else to add other than this wouldn't have been possible without that collaboration within the scientific communities, as well as the enormous funding opportunities that were available due to this huge problem.
Thanks. Dr. Keefe, you touched on a moment in your intro about the subscription rates to vaccines historically. And recently we've seen a lot of rise of the anti-vaccine movement, that sort of thing. In large part because of the fear of the side effects of these vaccines and whether in fact they cause autism or not. Now, I think it's pretty clear from all the data that we have that vaccines do not in fact cause autism, but I wanted maybe if you could speak to the history of this sort of vaccine, anti-vaccine movement in the relative to the other vaccine efforts.
I can say a little bit about it. I basically don't understand it, but there was a study in the 1990s, I think, that suggested that, I believe it was the then whole cell whooping cough vaccine, that there was an incidence of neuropathology, perhaps linking to autism. That was thoroughly refuted just a very few years after that. At the same time, they changed the pertussis vaccine from a whole cell vaccine to a component vaccine, where components of the whooping cough bacteria are used in the vaccine. And everybody gets it's the DTaP vaccine, diphtheria, tetanus, and acellular pertussis. And I think there continue to be no credible data suggesting a link to autism.
I might mention it's that same ability to communicate quickly and efficiently that potentially has other side effects, right? The scientists were able to share their data and build on each other's work. Similarly, people who have a far lesser understanding of science perhaps were able to use that same internet technology to spread rumors and falsities. And perhaps because the world is evolving so fast, technology is moving forward so fast, it is challenging for anyone who is, less than an expert to really make a verifiable assessment themselves about something that is as consequential as vaccination. So I think it's really unfortunate that this movement against vaccination has taken such a strong hold. One certainly can point out the fact that some vaccines are more effective than others. And that has as much to do with the biology of our bodies as it does with the technology that's eliciting an immune response. But in terms of negative consequences, well, I think we'll all agree that nothing is absolutely 100% effective or without any danger. That has to do with, under all circumstances with a human being. But as Dr. Chakarian was pointing out, these studies were done really carefully with a lot of people and looking out for potential adverse reactions. And I will cover some of that, and as will Dr. Turner. So perhaps we could move forward.
Yes, just one last piece of information I'd like to present. Thank you, Dr. Rowe, for that. And what you mentioned really spoke to me in the last few months, because this saying of lies travel around the earth 8 times before the truth has the chance to get out once, right? Well, to that effect, and thank you for those of you that responded to the Google form. We got some interesting responses there. And I think this applies just generally broadly, but essentially everybody said they had heard one of the following myths from somebody that they trust. COVID is a hoax, that the vaccine won't help you get immunity from the virus. The pandemic is a means of governmental control. Vaccines cause other diseases like schizophrenia and autism. The vaccine has harmful ingredients in it, so on and so forth. So these are lies perpetuated by people that we trust. So I don't think anybody has been immune from that during this pandemic. Is that we get people that we trust having opinions, and those opinions travel much faster than the scientific community can keep up with, certainly. So thank you.
We should remind ourselves that trust is a contextual concept, right?
Yeah, definitely.
You cannot blanket trust expertise to anyone.
Yes, agreed. Okay, so with that, why don't we turn it over to you, Dr. Rogel, and we'll listen to a little bit about the molecular biology and the biology behind the vaccines. How do they work?
Okay.
How do they elicit an immune response for us?
Thank you. I will share the screen. All right. So, you know that when we speak or sing or cough or sneeze, we expel droplets of our saliva, droplets that still contain everything that is in our respiratory system. These droplets very much vary in size, and as such, they also vary in how long they remain hanging in the air around us. A good sneeze will travel at some 50 meters per second, and before it falls to the ground, usually those are the big droplets, go as far as 18 feet away. Now, when the sneezing, coughing person is infected with a virus, for example, a SARS-CoV-2 virus, those droplets will carry the viruses. And the viruses, just before the virus concentration, just before the symptoms set in, and for a couple of days thereafter, are at a very high concentration. We're talking about 1 million viruses per mil. And yes, they travel with the sneezes and the coughs and the conversations and the singing. And they also hang out in the air in these aerosol, tiny little aerosol droplets, sometimes for hours. So that when you come along and you inhale that aerosol, inhale those droplets, they enter your nasal passages and they enter your lungs, where they settle onto the surfaces, and the viruses therein come into contact with cells that line those passages and engage in a binding with the cells through their receptors. Now, it's these spikes that engage into what's called an ACE2 receptor that is present throughout the body. And through that interaction, the virus is internalized and it opens up inside the cell and initiates the replicative process inside the cell. Now, what we should remember is that this is an RNA virus, a virus which contains, whose code to build more viruses is written in RNA nucleic acids. Okay, so that once the RNA is found in the cytoplasm, that RNA can immediately be translated by using cellular machinery into many, many different proteins that it contains. Among them are the spike proteins that we're going to be focusing on. But it takes all those proteins in order to then reassemble inside the cell another virus and release it into the environment. Now, I might point out that it's not one virus in and one out. What virologists like to say, one virus in and hundreds out. Now, when that happens, the viruses then are spilled into the environment and they infect other cells and infect other tissues, and the immune system kicks in. And the immune system is a complex process, which takes time to develop, but it is also very versatile, and it can develop very specific and long-lasting immunity to whatever pathogen it encounters. It's got two arms, and I'm going to tell you about that in a minute. It's got a cellular side to it and an antibody side to it. But the thing that we want to remember right now, this process of developing immunity takes some time. Now, while the immune system is gearing up to develop the antibodies and cells that recognize infected cells and kill them, the virus spreads throughout the tissues of the body and infect, yes, of course, the lungs, as well as the brain. That's why we see the side effects of changes in smell and in taste. Liver gets infected, as do the pancreas and the heart and the intestines. The infection of the intestines accounts for the diarrhea that one is seeing. And now all along, so while these infections are taking place, the viruses are causing damage to the cells within these tissues, within these organs. The virus, given its receptor, is causing blood volume dysregulation, and all along, all these tissues are screaming for help and sending out help signals that we know as inflammation, and you have heard being called a cytokine storm. So you get this enormous amount of damage not just in the lungs that are mostly talked about, but in fact throughout the body, from through kidneys and various and the heart. Okay, and that clearly has an impact on the individual. Not all that are infected develop this this pathology. In fact, as many as half of the infected individuals do not have any symptoms, but the ones that do can get really, really sick. And of the people who become symptomatic, the majority of those individuals has been younger than 65 years of age. However, the death toll of this viral infection is primarily among those that are older than 50 years old or older. In fact, 95 Americans that were killed by COVID-19 were 50 years old or older. So the question is, how do we prevent this acute SARS-CoV-2 infection? What we want to do is pre-alert and train that immune system in order to be ready for when the real virus comes along. And in order to understand that, we want to see how the immune system works. It is complex, it is a multi-step process, and I will try to be as straightforward with it as possible. But when a virus, which we often call an antigen, which we can call an antigen in the context of the immune system, infects a cell or is engulfed by a particular kind of a cell that monitors our tissues for unknown pathogens, that antigen presenting cell calls in for help sorry, my mouth is dry. So upon an infection, the antigen presenting cells show bits of the virus on their surface and present it to a different kind of a cell, the white blood cell that's called a helper T cell. And that helper T cell calls in other white blood cells, which then reach out towards these two approaches of dealing with the virus. Helper T cells will activate another white blood cells called the B cell into producing antibodies, and those antibodies, once they are found in plasma, released into plasma, can surround the virus and prevent it from binding to a potential target cell. That same helper T cell will also engage with a different kind of a cytotoxic T cell. That cell, upon activation, becomes a killer cell, one that recognizes an infected cell and causes it to die. Also, at the same time, while these antibodies are being produced and cytotoxic T cells are being activated, some of the cells, some of their members, set aside and go into hibernation and become memory cells, cells that remember how to make antibodies and cells that remember how to make more cytotoxic T cells. And they sit around until the second exposure of that same pathogen, that same antigen. And when the second exposure occurs, instead of starting this whole process all over again, the memory cells can be awakened and bring about a really quick response, very quick production of antibodies that are released into plasma and can coat the free virus and block it from infecting cells, as well as an increased production of these cytotoxic T cells that recognize and directly kill the infected cells. And of course, more memory. So this slide here, or this picture here, this diagram here says exactly the same thing. And I put it there just to reiterate the fact that once you have the initial exposure to a pathogen, the immune system cells talk to each other, triggering two branches, two kinds of responses. One that ends up in formation of antibodies, those are found in your plasma, in your blood, and another kind of white blood cells that can directly attack the infected cells. Plus, you have your memory cells that, upon second stimulation, can jump into action far more quickly. And in order to address the issue as to how quickly one is protected once immunized, I think it's best to look at how quickly antibodies, which accumulate in response to either to an infection, how quickly they are produced. Suppose you have the first exposure to a pathogen on day 0. It takes 2-3 days before all these cells can start producing significant quantities of the antibody. But then very quickly, the antibody concentrations start decreasing. And over a period of time, they go to very low, almost undetectable level. However, if you come in some 2, 3, 4 weeks at the earliest with another exposure to that very same pathogen, to that same set of epitopes, antigens, that those memory cells can awaken really quickly and start pumping out both the killer cells as well as the antibodies, and do so in a much larger quantity. So you get this much increased response to a booster, if you will, infection, second exposure to the antigen. Now, what we want to do is protect a person without actual exposure to the virus. And one can do that in many different ways. One of them, sort of a standard one that Dr. Kieft referred to, is an inactivated pathogen, a virus that has been killed, a virus that still shows all those viral-like shapes on its surface, but actually is incapable of taking over a cell. We can also isolate some typical proteins from a virus and inject those and teach the immune system to make antibodies and an immune reaction to that. Or we can create virus-like particles from bits of viruses. We can also take bits of the genome from the original virus and incorporate it into another virus that by itself does not replicate inside the cell, but is capable of infecting it very efficiently, or and then cause the synthesis or bring about the production of the proteins from the original virus. We can also just introduce the original RNA or DNA, but those tend to not be very stable by themselves. And yet, all these approaches have been tried in a variety of for a variety of different pathogens, and we don't have time to go through them right now. But there are many different ways of developing the material that then will function as a vaccine. But each one represents has its pluses, some have their minuses, and some are technological, and some have to do with having a different kind of an impact on a human being. So I'm just gonna show you this map of the different viruses that are being developed throughout the world right now, and we will focus on the vaccines that we have now available, or soon will have available here in the United States. And I will focus on the RNA vaccines of Moderna and Pfizer, and I will tell you a little bit about how the protein subunit vaccine, like the Novavax, would work, as well as the viral vector Johnson & Johnson vaccine would work. So here we have our SARS-CoV-2 virus, and we want to build an immune response to it without actually infecting a person. And COVID is a coronavirus and is coated in these proteins, this lipid membrane that contains these spike proteins that I'm sure you've heard about. And those spike proteins are the ones that engage with cellular receptors and allow for the virus to be internalized. They're also the most visible to the immune system. It's the first thing that the immune system will see is all those spike proteins assembled on the virus itself. Now, those spike proteins are encoded by viral messenger RNA, just a fraction of the total messenger RNA that is contained with the virus and encodes for how to synthesize more viruses in a cell. So just that fraction of a protein. And so what one could do is focus on the messenger RNA that encodes only for this spike protein and use it for production of the material that is to be injected into a person so as to elicit the desired immune response. So this is a super messy slide. I realize that. And there are things here that I won't be talking about, but I want to go through it step by step so that it makes sense as to how, in this case, the Pfizer vaccine would work. So here we have our virus, our coronavirus, SARS-CoV-2 virus. It's got a full genome, but only a fraction of it contains the code for the spike proteins that is required to infect you. So what scientists have done is taken the gene for the spike protein, pulled it out, and amplified it, and made large quantities of it, also modified it a little bit, so as to make it more stable. And then they wrapped it into a protective lipid capsule, so it cannot be readily degraded once it is injected into your arm. But once these lipid particles are presented to your tissues, they are internalized in a manner that is very similar to the way many other particles inside the body are internalized. And upon internalization of that messenger RNA, the cellular machinery translates that messenger RNA into the spike protein and makes thousands of copies per cell. Now, those proteins end up on the cell surface and are recognized by a different kind of a cell, by these dendritic cells they called. They essentially guards that live nearby, and they're constantly looking for any unusual shape that they have not seen before. And when they encounter such a shape, they engulf it and present bits of it on their own surface, so that these traveling immune cells called helper T cells that patrol our tissues throughout the body, when they encounter it, and if they recognize it with their own receptor shape, they become activated. And that is a signal that there is something new that's happening in our body. There's some new shape that could belong to a virus that is being produced nearby. And so once the helper T cells are activated, they bring in additional help. They encounter a different kind of a white blood cell that has receptors on its own surface that match the shape that they have recognized from the antigen presenting cell. And so the cells that match the shape are encouraged to multiply very quickly and into very large numbers, and differentiate into plasma cells that secrete huge quantities of antibodies right into our serum, so that when the virus is again found in our serum. It can be coated and prevented from binding to tissues that it otherwise would infect. Now, at the same time, that helper T cell activates the other arm of the immune response, the cellular immune response. It activates another subpopulation of cells called killer cells. And when after cross-checking that it's activating the right population with the right receptors on the surface, that activated cell is capable of encountering an infected cell recognized through display of the same spike proteins on its surface. And it's the killer cell literally causes the infected cell to commit suicide. So this expression of the protein, of the spike protein on the surface of an infected cell is translated into activation of the immune response and into immediate production of some antibody and some activated killer cells, but also these memory B cells to make antibodies upon second exposure and targeted T cells upon re-exposure, right? So Pfizer vaccine requires 2 doses, requires the first vaccination and then a booster 21 days later, and so as to give you this very strong protective response in terms of both antibodies as well as the cellular response. So you become protected only 2 weeks after the second dose, you are considered protected. And when Pfizer was doing the clinical studies, and Dr. Turner will tell you more about that, they got data that it was absolutely amazing. Could not be more obvious that the vaccine is protecting the vaccinated groups, whereas in fact, they had enrolled something on the order of 40,000 volunteers and gave them the first dose, gave them the second dose, and by the time the 4 months were over, about 170 people had gotten infected in the placebo group, whereas only 8 in the vaccinated group. So fantastic outcome, better than most other vaccines. Now, most of us will be getting Moderna, and that also is a 2-step vaccine based on on highly analogous mRNA technology. Some of the details are different. The sections of the RNA that are used to vaccinate with are a little different between Moderna and Pfizer. The packaging is a tiny little bit different, but it all works more or less the same. The vaccine particles are taken up, mRNA is translated, the proteins are recognized by the dendritic cells. They activate the T helper cells, which in turn bring in the 2 arms of the immune system and generate a long-term memory. And again, the timeline, because it's based on the biology of these processes, is very similar, and you're considered to be protected about 2 weeks after the second exposure. The Johnson & Johnson vaccine is a little different. The immune response ultimately is very similar, but the vaccine itself structure is based on an adenovirus, another virus that most of us have had at some point because it causes cold, but it's been modified to the point where it can infect, so very efficiently introduce nucleic acid sequences into the cells, but itself cannot replicate. So what Johnson Janssen Pharmaceuticals did, they converted this spike protein RNA sequences into a double-stranded DNA sequence and packaged it into an adenovirus, which now is very efficient into introducing that DNA into the cell. And because it's DNA rather than RNA that is now being now serving as a template, you get many more copies of messenger RNA and more copies of a protein, which means that the whole process of immune stimulation needs only a single dose. You get a continuous transcription of the messenger RNA of this double-stranded DNA, and you get ultimately a very good response. And it's been this Johnson & Johnson vaccine has been demonstrated to provide complete protection against hospitalization and death. So about 28 days post the single dose vaccination. And we are waiting any day now. We should be where FDA approval is expected for this vaccine. And then there's another one that's likely to become available in the United States, and that's a Novavax vaccine. There, the spike protein is synthesized in insect cells, and it's synthesized in such large quantities that you can purify it and assemble, sort of aggregate it all together. And when you add sort of a stimulant of the immune system called an adjuvant to it, it's those particles now that have no genetic material are taken up by those same dendritic cells that ultimately express it on their surface, recruiting the helper T cells that then recruit the two arms of the immune response and eventually leads to this robust response about 2 weeks after the second dose. Now, I promised to offer the contents of the Pfizer and Moderna vaccines. It's actually, as these things go, super simple ingredients. We have the messenger RNA, right, the segment of the RNA that encodes for the proteins and is ultimately translated into the proteins that trigger off the whole cascades. Lipids are there. They look, those names of chemicals are very fancy, but they're essentially the same kind of lipids that surround every cell of your body. There are some salts to balance the salinity and acidity, as well as some sugar to help maintain the shape during the freezing. Sucrose is the sugar that is used. Now, Moderna has essentially those very same components. There are some minor differences, and I've highlighted them here. One of them is, and I've highlighted the chemicals that could be of some potential concern. One of them is polyethylene glycol. It's present in both of these vaccines as part of the lipid coat that the RNAs are wrapped in. And it is thought that they are the ones that may be the cause of the very rare anaphylaxis that has occurred. I want to put that in perspective. The observed anaphylaxis with these two vaccines has been 2 to 5 people out of the million that have been vaccinated. And that is to be compared with 1 person per 5,000 people that experience anaphylaxis upon vaccination with the flu vaccine, which is far less efficient. So, this is clearly getting some attention. The other sort of unusual thing here is in messenger RNA. In order to make this messenger RNA more stable and better translated, some unusual nucleotides have been incorporated. There have been some replacement of uridine to pseudouridine, and I want to emphasize that stabilizes the RNA, but it can at no time change your genes or get incorporated into your DNA. So, having said too much already in too much detail, I'll turn it back to you.
Thank you, Dr. Rogel. That was so helpful for going through everything, and especially the different activities of the different viruses. Do any of the other panelists have anything to add? Especially Dr. Chakarian now?
I think that was a really fantastic overview of the immunity of these vaccines. Thanks.
I agree. And thank you to Dr. Kieft and Dr. Chakarian for answering those questions in the Q&A as well. One thing that that I think passed my mind as maybe a source of confusion that you alluded to just now is that these RNA viruses, or these that the RNA vaccines rather,
Mhm.
are not changing our genome.
Mhm.
So RNA does not integrate into our genome. Is that correct?
That is correct. To the best to our knowledge, there isn't a mechanism whereby these messenger RNAs could integrate into our genome and stay with it. The RNAs themselves are degraded rather quickly in the cytoplasm, and that's why those modifications had to be incorporated to make them longer lasting, to serve as a longer lasting template to make more copies of the protein and then amplify the response.
Great. Okay, that is good news.
Yes, very good and very clever.
And perhaps part of the reason why there's 2 doses rather than 1, in that, as you mentioned, with the Johnson & Johnson vaccine that produces a long-term attenuation of the immune response, but because the mRNA is degraded so rapidly, it doesn't stick around, so you have to get that second dose.
That's right. Whereas the DNA can stick around for a prolonged period of time, and it sort of serves as its own booster because it continuously produces that messenger RNA. There are some vaccines out there that are also viral adenovirus-based vaccines. In fact, the Russian vaccine has 2 shots, and they both DNA viruses. And so that's just another approach. But it seems that for Johnson & Johnson, a single dose provides the efficacy that has been reported.
This is a little bit of a technical detail, but another advantage of the mRNA vaccines is that you probably know your DNA is inside the nucleus of your cells, okay? And these RNA vaccines, they actually don't need to go to the nucleus in order to start making the proteins that induce the immune response. So that's another sort of level of safety, I think, of these vaccines, is they don't actually go anywhere near, where your genome is, where your DNA is.
Yeah.
Great, thank you. That is a nice lead in then, I think, to Dr. Turner. Dr. Turner, go ahead and share your screen and take it away.
Will
wait,
do.
one thing quickly, sorry. I hit the wrong button here and I meant to type this answer, but yes, this recording is currently live on YouTube and will be made available and stay available permanently. I will send a link tomorrow with the recording data. So yes, thank you, Vicki Conley, for that question. And with that, Dr. Turner, go ahead and take it away.
Great. And before I do, I saw you, Dr. Kieft, speaking, but you were muted. I don't know if there was something you wanted to say before I get started.
Oh, why don't you go ahead, Danielle,
Okay.
and if there's time, I'll mention it later.
Got it. I will share my screen. Perhaps, there we go. Okay, so I don't know about you all, but I am completely in awe of our immune systems and the technology that these mRNA vaccines are offering and the protection. But I just want to touch on the 4 vaccines that are currently available. As you guys have probably heard of, we've got the Oxford AstraZeneca, our Moderna and Pfizer vaccines, as well as the Sputnik vaccine. Just so we're aware, there are other vaccines out there globally, but today I'm just going to focus on the Moderna and Pfizer mRNA vaccines because that's what is available to us currently in the US. So there's a lot going on in this slide. As they would say in med school, this is a high yield slide because there's a ton of information in a really small space. So if you want any high yield information on these vaccines, this would be the slide to capture. But what I really just want to point out before moving forward is that these are very similar vaccines, as Dr. Rogel mentioned. They're both mRNA vaccines. They both stimulate the immune system in a similar way, and they're both about 95% effective. And as you all know, they require 2 doses to be maximally effective. But what I want to point out here, hopefully you can see my mouse.
We can.
great, is that after the first dose, and when I say after, I don't mean immediately after, it's about 2 weeks after, as we saw, it takes our immune system a little bit to ramp up and start producing protection from these antigens. Pfizer vaccine is about 88.9% effective, and the Moderna vaccine is about 80% effective. So after your first dose, you do get some protection, but again, it's important to maintain social distance and wear your mask because these studies were done on symptomatic COVID cases. So we're still not entirely sure if you can still pass on COVID or harbor it after you get the vaccine, but you will not get sick. At least 95% of you will not get sick or have any type of symptoms. So going a little further, I just want to reorient us and make us all familiar, put us in a familiar place with vaccines that we're used to. So we've got our flu vaccine, and we're not going to talk a lot about it because I could get into a ton of details and we just don't have time. But for example, this is an inactivated viral vaccine. So that means you've got the vaccine, it's grown up, and then it's killed and it's injected, and your immune system recognizes it as a whole particle. Whereas you've got your MMR vaccine that Dr. Kieft introduced really nicely. And this is so effective. It's essentially, other than our COVID vaccines now, the most effective vaccine that we have, 97% protection. And that's because this is a live attenuated vaccine. And what does that mean? That means essentially you're taking the virus and you're making it to where it's less infectious, but it's still alive. So your body is thinking that it has an actual infection and it's mounting that response and kicking off every aspect of your immune system. And so these are, you know, the most effective vaccines. But what's incredible, and I'm amazed by it, is that we've got this, you know, 95% efficacy with mRNA vaccines. It's just a code, as Dr. Rogel really nicely demonstrated. Our cells take up that code and they produce that one little protein. So you don't need a live vaccine. And what's even better about that is that the risks are very low, meaning you will not get infected by Pfizer or Moderna. You cannot get COVID. Your cells will not produce COVID, is what I should be saying, when you get this vaccine. They will not produce a particle or a virus. They'll just produce that one little spike protein that your immune system will recognize. Whereas, as amazing as this MMR vaccine is, you know, 97% efficacy, there are risks that it can revert back to its more infectious form. There are, you know, cases of the polio vaccine, which is live attenuated, that have reverted back to its infectious form and given people polio. That is not at all a risk with these mRNA vaccines. So they're incredibly safe. So there were a lot of questions regarding how the efficacy is calculated, and I too was curious. We will go through a beautiful New York Times article, consulted with CDC, and put out really nice step-by-step calculations that we'll go through together. This is for the Pfizer data for their clinical trials. So what they did is they had about 43,660 participants. And of those 43,660 participants, they were broken up equally into placebo or the group who received the vaccine. After some time, 162 people who received the placebo were infected with COVID, and only 8 people who received the Pfizer vaccine contracted COVID. So in order to figure out the infection risk, you just take 162, which is your number of infected people, divided by the total group size, and you get an infection risk of 0.74%. You do the same thing for the number of people who contracted COVID when they received the Pfizer vaccine, and you get 0.04%. So what does this mean when we scale it up? We scale this up by saying that there are about 328 million people in the United States. And of those people, how many would we expect to contract COVID when they don't get the vaccine? So you do that by multiplying their infection risk of the placebo group, the 0.74%, by the 328 million population in the United States, which leads to about 2.5 million infections when no vaccine is administered. And you do the exact same thing with the infection risk related to receiving the vaccine, and you get about 131,000 infections when you use that risk. Okay, so what does that really mean for efficacy? These are just a bunch of small decimal numbers or large population numbers. So the next thing that you want to do to find the actual efficacy of this Pfizer vaccine is you take the difference between the risks in both groups. So 0.74% risk of infection when you don't receive a vaccine and 0.04% risk of infection when you have gotten the Pfizer vaccine. And this risk is 0.7%. So this is saying, you know, the vaccine reduced the infection risk by 0.7%. And this seems like a really small, insignificant number, and I was not wowed by it. But then I read further. Okay, so how does this translate into the 95%? So you do this by scaling the percentage point difference. So you take, you know, there's a 0.7% difference in your risk between vaccinated and unvaccinated. And you divide that by the total risk of unvaccinated people. And that's where you get your 95%. So under the same conditions as the study, the Pfizer study that we showed on the last few slides, the vaccine reduces the risk of infection by 95%. So that 0.7% difference, which seems really small, actually translates into a large difference in the number of people that become affected with COVID. And this 95% efficacy, again, we were all excited about this. This is really incredible. We're not using a live attenuated vaccine. We're using mRNA, just a transcript. But that 95% effectiveness does translate into, you know, you're going to get side effects, as we all learned from Dr. Ruggel. When the immune system responds, you're going to feel it. These mRNA vaccines are reactogenic, meaning that they do stimulate a strong immune response, which leads to these side effects. So what are they? In Pfizer, out of the 36,600 people, main side effects: injection site reaction, fatigue, headache, muscle pain, chills, joint pain, and fever. Again, none of this is caused by your cells producing virus. What this is caused by is your cells recognizing that one spike protein and sending off a cascade of events. So your own body is creating the symptoms. It's not a virus, you're not infected. And for Moderna, they are very similar. Injection site pain, fatigue, headache, chills, joint pain, and muscle pain in many people. And these side effects are after the second dose, more often than not. And just side by side, again, after the second dose. And why does this occur more often after the second dose? Again, going back to that really nice graph showing the response of the immune system, the first dose of a vaccine serves as a primer. So it's telling your body and your cells what to look for. Once they start eating that little antigen, that spike protein, they remember it, as Dr. Ruggel said. So the next time that they're exposed to that same spike protein, they recognize it, they've already mounted their little army, and then they respond vigorously. And so that's why your body produces these symptoms, because your immune system is doing an amazing job recognizing these spike proteins and attacking them really quickly this time. And I can say from experience, I received the Moderna vaccine about a month and a half ago. After my first dose, my arm was sore for a couple of days, annoying, but nothing, you know, groundbreaking. And then I felt fine. After my second dose, I knew that my immune system was doing a really amazing job, and I felt pretty crummy for a couple of days. I got a fever, muscle aches, I was tired. But again, a couple of days, knowing that your body is doing what it's supposed to, knowing it's mounting this response felt, you know, really great, even though I felt really crummy. So I tried to imagine, I'm feeling horrible now, but this is 2 days. I can't imagine getting an actual COVID infection and feeling like this for 10 plus days, or even having lingering side effects for, you know, months. So I would say be prepared when you get this vaccine, but these are really small annoyances. It's not bad at all, and everyone responds differently. So it's just your body doing its job. So how long does it take the COVID vaccines to work? So both Pfizer and Moderna. As I mentioned, day one, you get your first shot. A lot of people feel a bit of pain in their arm. Some people report feeling fatigued after the first shot, but it's really rare. And if you do, that's okay. Take some Tylenol, and that typically subsides, your pain subsides. And then a couple weeks later, when your body starts mounting its response, you can have, the 80% coverage. But as many experts say, keep wearing a mask and social distancing, do your part in slowing the spread of COVID. And then for Pfizer, 21 days later, you get your second shot. For Moderna, 28 days later, you feel crummy for a couple of days. And 2 weeks after that, you are, 95% protected, which I think is a really good trade-off for feeling crummy for a couple of days. And again, not everybody feels crummy. Maybe your arm's just going to be a little bit more sore. So I'm not going to go into a lot of detail on this because Dr. Chakarian described it really well. So here's just a visual to what we were talking about earlier, which is, typical vaccine development is 5 to 7 years for clinical trials, whereas for these mRNA vaccines, we got them done in a few months because we have collaboration. This is pre-existing technology, and the huge money that was thrown at developing these to help curb the deaths from COVID and the infections from COVID allowed these studies to be run in parallel. So nothing was skipped. It was just sped up. And that's all I have.
Thank you so much, Danielle. That was wonderful, wonderful presentation. Thank you for going through the efficacy and how that's calculated and the safety data as well. Do any of our panelists have any additional contributions they'd like to make to safety and efficacy?
Well, I'll just say something I was going to say before, and it fits right in with what Danielle was talking about right at the end there. So Dr. Chakarian gave us a really good rundown of how it was possible to develop and also test the vaccine so rapidly. And one of the factors is mRNA is really easy to make. It's made from DNA. Most of us don't synthesize our own DNA, but we can order it up, and I can type a sequence of nucleic acids into my computer and have DNA delivered to my lab the next day. And then it's a simple enzymatic step to make messenger RNA. So it's how they could make tons of it. The technology was already there. It was already being developed as a virus, as a vaccine, as we heard. So it was just a matter of adapting it to SARS-CoV-2.
If I can say one thing about molecular biologists, we are master stealers of what exists, be that in nature or out wide. That's what we do for a living, is steal the technology that exists and apply it to new problems and questions. And I think this is definitely a very good example of that. Dr. Rogel, you were about to say something.
Well, I agree with you completely. Incredibly good thieves we are, and recombiners.
That's the word I was looking for, not stealers. Yeah.
Yes, no, I like that.
Not good wordsmiths.
Especially. But I was going to sort of extend Dr. Kieft's comment because it is so easy to synthesize, to design a piece of DNA with a particular sequence, which then can be very readily made into millions and millions of copies of messenger RNA, which then immediately get translated once they're in the cytoplasm with this translational machinery that every one of our cells has. It also speaks to the potential of this vaccine in terms of adapting it to the new variants that are coming up, right? The major differences in those variants lie in the sequence of this messenger RNA, which ultimately is translated into an ever so slightly different protein. And while it is complicated to produce large quantities of either inactivated viruses or the spike protein itself, what is relatively easy to do as anything goes is that instead of encapsulating today's most commonly infectious COVID variant into the vaccine, one only needs to tweak the nucleotide sequence, and that ultimately will change the amino acid sequence that is being presented to the immune system and serve as a booster to the pre-existing current vaccine.
Thank you, Dr. Rolo. That's something that that a lot of scientists are concerned about, is the variants that are arising, like the South Africa variant or the UK variant, so on and so forth. But as you just said, we have the ability to tweak our current vaccine to suit those variants. So while we may not have that capability to combat those variants immediately, that capability exists such that we can scale up as needed. Is that right? Is that your understanding?
Yes, that is definitely my understanding, and we can also pivot in terms of addressing slightly different variants as they will have naturally evolved for sure.
I think the FDA announced today that any new mRNA vaccines that are based on variants won't have to go through the whole clinical trial process. They can go immediately to people. So it just has to do with the really stellar safety data of these vaccines and also how strongly immunogenic they are. There was a question in the Q&A about sort of asking about our experience with mRNA vaccines, how they had been tested before, how they gone into people. That I thought was a really good question. So Moderna actually is a company that is built around developing mRNA vaccines. And so they've developed a whole bunch of different mRNA-based vaccines for influenza, for cytomegalovirus, for respiratory syncytial virus, lots of common pathogens. And some of those have actually gone into early phase clinical trials. So there was some safety data, not a huge amount of safety data, but there was some safety data from those Moderna trials already. I think, for scientists, I think the main question we had going into the mRNA vaccines was not, are they going to be safe? I think we all felt pretty good about the safety of these vaccines. I think the concern was, are they going to induce strong enough immune responses to provide protection against the virus? And that was the biggest question that, as a vaccinologist, that I had going in, because there wasn't a lot of data on whether these vaccines induce strong immune responses. And the answer is clearly that they induce really, really good immune responses. And it turns out that they actually induce better immune responses against the coronavirus than getting infected with coronavirus does, which is really quite impressive. There were a couple of questions in the chat asking about that. And there may be probably a number of different reasons for that. But again, I think these vaccines have really surpassed everyone's expectations on how well they would perform.
I ask a question on that. We still have a lot of diseases for which there are no vaccines. Are there some candidates out there for application of the messenger RNA approach that might be near term even?
Yeah, it's a good question. So the way I tend to answer this is that there are easy vaccines and there are hard vaccines. Okay, so when you think of something that it's been very difficult to develop a vaccine of, think of, for example, HIV. There's a lot of reasons why it's been very difficult to develop vaccines for HIV. There's lots of different strains of HIV. An antibody that protects you against one strain doesn't protect you against another. So there are features of that pathogen and other pathogens like it that can make it very, very difficult to develop vaccines. For coronavirus, it appears, at least right now, to fit into the easy category, where we're using a technology that's new, but there are more established technologies that are still in development in clinical trials that could work just as well as these mRNA vaccines. So I think this might be a case where this was an easy vaccine to make. Now, as the virus evolves and our abilities to develop vaccines that keep up with, variants of the virus, whether that's going to be a problem or not, we don't know the answer to, but that could complicate things slightly. But I think that's sort of the short answer. This is an easy vaccine.
Thanks.
Thank you. I had one final question for any of the panelists, if you don't mind. If anybody knows, I think we're all interested to see. I think pretty clearly the vaccine protects us from severe disease. Do we have any recent data indicating about how the vaccine affects transmission of the virus? Snezna.
Maybe I can start and Bryce, you can add to it. But to the best, so the trials were carried out with symptomatic as being an infected person. Now we do know that one can be asymptomatic, but still carry the virus. So I just want to point that out, that was the measuring stick there. But once one is vaccinated, it is my understanding that one still can get infected, right? That one can still be infected at a very low level. The result being that you are producing much fewer viruses, which have less of a chance to spread to other tissues, which have very, very, very much lower chance of causing any kind of pathology, and in fact, much lower chance of becoming symptomatic. However,
I think we lost Dr. Rogel.
You just froze.
I think,
Probably fill in what she was going to say. So
yeah, go ahead. I was
However,
thinking that you could.
you're still at a risk of transmitting to somebody else. Actually, I think some of the most interesting data on this has just emerged in the last week. So the country of Israel actually has very high vaccination rates. And so they've actually just started generating data on transmission by people who have been vaccinated. And again, this is very early preliminary data, but it seems to suggest that getting vaccinated reduces your chances of transmitting to somebody else by about 90%. Okay.
Oh, wow.
So,
Awesome.
so that is really, so I was just mentioning the Israeli data that just came out, for those of you who just came back on. And so that data is really encouraging, that vaccination is also going to prevent transmission. Again, it's not like there is no risk. There's a small risk, just like there's a small risk of getting a symptomatic infection after you've been vaccinated. But again, I think it's very encouraging that, that the number is so high. So that was with the Pfizer vaccine. So we'll just have to see, how the other vaccines perform.
Great. Wonderful.
if I could just add something, sorry,
Yes, please.
computer decided to check out for a bit. What was also shown is that, of course, once you're vaccinated, if you are infected and you do produce some viruses, the tighter the number of viruses that your cells are making is way, way, way lower, right? So not only does it make you less sick, what you are transmitting, the sheer density of viruses in your saliva are way lower. And what has been shown is that the initial inoculum matters. If you're inoculated with fewer viruses, you're less likely to get very sick than if you're inoculated with a very large dose, infected with a very large dose. So you are less likely, if you yourself are producing fewer viruses, you will less likely to make another person very sick. Now, of course, susceptibilities vary from person to person, but in general, the number of viruses that you have been exposed to contributes to the severity of the illness. So a vaccinated person will give you fewer viruses. And that's why we're continuing to recommend that the face masks should be worn and distances should be abided by.
Wonderful. Okay, so I think overwhelmingly the conclusion is get vaccinated and continue to be careful following vaccination. Would my panelists agree with that?
Yes.
All right. All right. Well, with that, I think that concludes our panel today. Thank you so much to our panelists who did a wonderful job. I will follow up with the recording of this tomorrow, as well as if the panelists agree that I can share their slides, I'll send their slides along as well. Just thank you for your attendance, everyone, and especially thank you again to our panelists who did just a a lot of extra work beyond their normal day jobs to prepare this information for you. So thank you so much, Dr. Rogel, Dr. Keefe, Dr. Chakarian, and Dr. Turner. Really appreciate your time and effort here.
Thank you for organizing this, Joel. It was a great idea. I hope it's helped some people.
Thanks so much.
Me
Thank
too.
you.
All right. Goodbye, everyone.
Bye.
Be well.